Today we will learn about immunity, innate and adaptive, cells, tissues and molecules of immune system. The immune system is a remarkably versatile defense system that has evolved to protect animals from invading pathogenic microorganisms and cancer. It is able to generate an enormous variety of cells and molecules capable of specifically recognizing and eliminating an apparently limitless variety of foreign invaders. The Latin term immunis, meaning exempt, is the source of the English word immunity, meaning the state of protection from infectious disease. The immune system includes two main components, innate and adaptive immunity. The innate immunity is less specific and provide first line of defense against infection. Most components of innate immunity are present before the onset of infection. The innate immune system, also known as non-specific immune system, comprises the cells and mechanisms that defend the host from infection by other organisms in a non-specific manner. This means that the cells of the innate system recognize and respond to pathogens in a generic way, but it does not confer long-lasting or protective immunity to the host. Innate immune systems provide immediate defense against infection and are found in all classes of plants and animal life. The major functions of the vertebrate innate immune system include: number 1, recruiting immune cells to the site of infection through the production of chemical factors including specialized chemical mediators called cytokines. Number 2, activation of the complement cascade to identify bacteria, activation of cells and to promote clearance of dead cells or antibody complexes. Number 3, the identification and removal of foreign substances present in organs, tissues, the blood and lymph by specialized white blood cells. Number 4, activation of the adaptive immune system through a process known as antigen presentation. Number 5, acting as a physical and chemical barrier to infectious agents. Innate immunity comprises 4 types of defense barriers: anatomic, physiologic, phagocytic and inflammatory. First we will see anatomic barriers. These include skin that provide acidic environment to retard the growth of microorganism and mucous membranes that entrap foreign microorganisms. The figure demonstrates the external defenses that act as anatomic barriers. The epithelial surface form a physical barrier that is very impermeable to most infectious agents, acting as the first line of defense against invading organisms. Desquamation of skin epithelium also helps remove bacteria and other infectious agents that have adhered to the epithelial surfaces. In the gastrointestinal and respiratory tract, movement due to peristalsis or cilia helps remove infectious agents. Also, mucus traps infectious agents. The gut flora can prevent the colonization of pathogenic bacteria by secreting toxic substances or by competing with pathogenic bacteria for nutrients or attachment to cell surfaces. The flushing action of tears and saliva helps prevent infection of the eyes and mouth. Physiologic barriers include temperature that inhibits growth of some pathogens, low pH, that is acidity of stomach that kills most ingested microorganisms, and chemical mediators with lysozyme, interferon, complement, colectin, etc. that mediate defense by various mechanisms. Phagocytic or endocytic barriers include various cells that internalize and break down foreign molecules and specialized cells like blood monocytes, neutrophils, tissue macrophages etc. that internalize, kill and digest whole microorganisms. As can be seen in the figure, the phagocytes mediate attachment of the bacterium to its surface, thereby internalizing it and finally leading to its killing and degradation. Inflammatory barriers include tissue damage and infection that include leakage of vascular fluid containing serum proteins with antibacterial activity and influx of phagocytic cells into the affected area. Inflammation is stimulated by chemical factors which are released by injured cells and serves to establish a physical barrier against the spread of infection and to promote healing of any damaged tissue following the clearance of pathogens. The process of acute inflammation is initiated by cells already present in all tissues, mainly resident macrophages, dendritic cells, histiocytes, Kupffer cells and mastocytes. These cells present on their surfaces certain receptors named pattern recognition receptors, that is PRR, which recognize molecules that are broadly shared by pathogens but distinguishable from host molecules, collectively referred to as pathogen associated molecular patterns, that is PAMPs. The adaptive immunity is a more specific component of immune system. Adaptive immunity is capable of recognizing and selectively eliminating specific foreign microorganisms and molecules, that is foreign antigens. The adaptive immunity requires the interaction between various lymphocytes and antigen presenting cells. B lymphocytes and T lymphocytes both are produced in the bone marrow, but there is difference in the sites of their maturation. These lymphocytes have the ability of recognizing the foreign particles that are presented by specific antigen presenting cells or APCs. Several macrophages, dendritic cells, B lymphocytes, etc. can act as APCs and present the antigens together with MHC class II molecules to the lymphocytes to aid their destruction through humoral or cell mediated branches of immune system. This figure is the overview of the humoral and cell mediated branches of the immune system. In the humoral response, B cells interact with antigen and then differentiate into antibody secreting plasma cells. The secreted antibody binds to the antigen and facilitates its clearance from the body. In the cell mediated response, various subpopulations of T cells recognize antigen presented on self cells. TH cells respond to antigen by producing cytokines. TC cells respond to antigen by developing into cytotoxic T lymphocytes, that is CTLs, which mediated killing of altered self cells, example is virus infected cells. Cells of the immune system include the following: B cells. B lymphocytes are produced and matured in the bone marrow. The major function of B lymphocytes is to develop into antibody secreting plasma cells following stimulation by foreign antigens of bacteria, viruses and tumor cells. Antibodies are specialized proteins that specifically recognize and bind to specific antigens that caused their stimulation. Antibody production and binding to foreign antigens is often critical as a means of signaling other cells to engulf, kill or remove that substance from the body. Now we will see T cells. T lymphocytes are produced in the bone marrow, but they migrate to thymus for their maturation. They are usually divided into two major subsets that are functionally and phenotypically different. T helper cells, which are also called CD4 plus T cells, are involved in coordination and regulation of immunological responses. They function to mediate responses by the secretion of lymphokines that stimulate or otherwise affect other cells involved in the immune responses. The second subset type of T lymphocytes is cytotoxic T lymphocytes, that is TC cell or CTLs or CD8 plus T cells. These cells are involved in directly killing certain tumor cells, virus infected cells, transplant cells and sometimes eukaryotic parasites. CD8 plus T cells are also important in down regulation of immune responses. Both types of T cells can be found throughout the body, most conspicuously in lymphoid organs with lymph nodes and spleen, but also in the liver, lung, blood and the intestinal tract. Natural killer cells are also known as NK cells, are similar to cytotoxic T lymphocytes. They function as effector cells that directly kill certain tumors such as melanomas, lymphomas and virus infected cells. However, NK cells, unlike TC cells, kill their target cell without need for recognition of antigen in association with MHC molecules. NK cells that have been activated by secretions from CD4 plus T cells will kill their tumor or viral infected targets more effectively. Now, we will see macrophages. Macrophages are important in the regulation of immune responses. Besides their role in phagocytosis, they may function as antigen presenting cells because they ingest foreign material and present these antigens to other cells of the immune system such as T cells and B cells. This is one of the important steps in the initiation of an immunological response. Macrophages stimulated by certain lymphokines exhibit increased level of phagocytosis and also secrete cytokines that modulate immune responses. Next is dendritic cells. Dendritic cells also originate in the bone marrow and function as antigen presenting cells. In fact, the dendritic cells are more efficient APCs than macrophages. These cells are usually found in structural compartments of the lymphoid organs such as the thymus, lymph nodes and spleen. However, they are also found in the bloodstream and other tissues of the body. It is believed that they capture and process antigens in lymphoid organs where an immunological response is initiated. Of particular interest is the recent finding that dendritic cells bind large number of HIV particles and may be a reservoir of virus that is transmitted to CD4 plus T cells. Tissues of immune system. Lymphoid tissue is a specialized form of reticular connective tissue. The basic structure of the stroma is reticular collagen fibers sheathed entirely by fibroreticular cells and a few other elements such as CT trabecula and blood vessels. The thymus is a notable exception to this stromal structure. Lymphoid parenchyma tissue consists of large number of mostly free cells packed among the elements of the stroma. The parenchymal cells are primarily lymphocytes and their precursors. Lymphoid tissue is defined as being loose, dense or nodular. Loose lymphoid tissue is found throughout many other tissues and consists of aggregation of lymphoid cells in a reticular stroma, the stroma predominating. Dense tissue is similar except that the lymphoid cells predominate. Nodular tissue is composed of very dense aggregation of lymphoid cells in the form of follicles. Some follicles have germinal centers which are sites of active production of B lymphocytes. Most lymphoid tissues are rather disorganized and occurs within other tissue types. Some are found in particular organs and glands such as bone marrow, thymus, lymph nodes and spleen. Bone marrow is soft tissue located within the hollow interior portion of bones. There are actually two types of marrow. Red marrow produces red blood cells, platelets and most of the white blood cells, while yellow marrow produces a few white blood cell types. Immune cells differentiate from stem cells in a stepwise fashion. The process begins with hematopoietic stem cells, and from there a single cell differentiate into a range of cell types including myeloid cells and lymphoid cells. Each of these can further differentiate into more specific cell types, myeloid cells into phagocytes and granulocytes and lymphoid cells into B cells, T cells and natural killer cells. Thymus is a small gland which lies behind the upper part of sternum, and it is within this organ that immature T cells go through a complex process called thymic selection. Once the process is complete, T cells are fully mature and begin circulating in the bloodstream. At this point, having never been activated by an antigen, they are referred to as naive cells. Lymph nodes are nodular aggregates of lymphoid tissues that are located along lymphatic tracts within the body. Recall that lymph is the filtered concentrate of blood plasma found in the interstitial tissues. Not all of the fluid that leaves capillary returns to the heart via veins. Some of the fluid that leaves capillary is concentrated and drained via lymphatic vessels. Spleen is an organ that brings lymphocytes together with antigens that may be circulating in the blood. Blood that enters the spleen flows through a vast network of channels that are lined with phagocytes and dendritic cells. These antigen presenting cells can then convey the antigens to lymphocytes in the spleen. Other lymphoid tissues. There are many other small patches of immune tissues dotted throughout the body, most of which is classified as mucosal associated lymphoid tissues known as MALT. This tissue is simply a series of small areas throughout the body where lymphoid tissue is concentrated, but not in a large enough amount to be classified as a lymph node. There are such areas in the gut, bronchus, nose, larynx and eye, as well as on the skin and in the vascular system. In addition, there are the Peyer's patches of the small intestine which facilitate the mucosal immune response, and the tonsils and adenoids which again are similar to lymphoid nodes. The tonsils help protect against pharyngeal and upper respiratory tract infections, while the adenoids provide protection against inhaled pathogens. Molecules of immune system include immunoglobulins, cytokines, MHC molecules and proteins of complement system, which we will study in detail. Immunoglobulins are glycoprotein molecules that are produced by plasma cells in response to immunogen and which function as antibodies. The immunoglobulins derive their name from the finding that they migrate with globular proteins when antibody containing serum is placed in an electrical field. The basic structure of the immunoglobulin is illustrated. Although different immunoglobulins can differ structurally, they all are built from the same basic units. First is heavy and light chains. All immunoglobulins have a 4-chain structure as their basic unit. They are composed of 2 identical light chains which are of 23 kilodalton and 2 identical heavy chains of 50 to 70 kilodalton. Second is disulfide bonds. In that, first is interchain disulfide bonds, the heavy and light chains, and the 2 heavy chains are held together by interchain disulfide bonds and by non-covalent interactions. The number of interchain disulfide bonds varies among different immunoglobulin molecules. Second is intra-chain disulfide bonds. Within each of the polypeptide chains, there are also intra-chain disulfide bonds. Third is variable and constant regions. When the amino acid sequences of many different heavy chains and light chains were compared, it became clear that both the heavy and light chain could be divided into two regions based on variability in the amino acid sequences. These are first light chains. Again, these are of two types, VL and CL. VL 110 amino acids and CL is of 110 amino acid. Second is heavy chain, which is of two types, VH and CH. VH is of 110 amino acids and CH is of 330 to 440 amino acids. Fourth is hinge region. This is the region at which the arms of the antibody molecules form a Y. It is called the hinge region because there is some flexibility in the molecule at this point. Next is domains. Three-dimensional images of the immunoglobulin molecule shows that it is not straight as depicted in the figure. Rather, it is folded into globular regions, each of which contains an intra-chain disulfide bond. The general functions of immunoglobulins include the antigen binding. Immunoglobulins bind specifically to one or a few closely related antigens. Each immunoglobulin actually binds to a specific antigenic determinant and can result in protection of the host. Second is effector functions, which include number 1, fixation of complement. Second one is binding to various cell types like phagocytic cells, lymphocytes, platelets, mast cells and basophils. Cytokines are low molecular weight regulatory proteins or glycoproteins secreted by white blood cells and various other cells in the body in response to a number of stimuli. These proteins assist in regulating the development of immune effector cells, and some cytokines possess direct effector functions of their own. Cytokines bind to specific receptors on the membrane of target cells, triggering signal transduction pathways that ultimately alter gene expression in the target cells. A particular cytokine may bind to receptors on the membrane of the same cell that secreted it, exerting autocrine action. It may bind to receptors on a target cell in close proximity to the producer cell, exerting paracrine action. In a few cases, it may bind to target cells in distant parts of the body, exerting endocrine action. The term cytokine encompasses those cytokines secreted by lymphocytes, substances formerly known as lymphokines, and those secreted by monocytes and macrophages, substances formerly known as monokines. Many cytokines are referred to as interleukins, a name indicating that they are secreted by some leukocytes and act upon other leukocytes. Interleukins 1 to 25 have been identified. The major histocompatibility complex, that is MHC, is a cell surface molecule encoded by a large gene family in all vertebrates. MHC molecules mediate interactions of leukocytes, also called white blood cells, which are immune cells, with other leukocytes or body cells. MHC determines compatibility of donors for organ transplant as well as one's susceptibility to an autoimmune disease via cross-reacting immunization. In humans, MHC is called human leukocyte antigen. The MHC gene family is divided into three subgroups: class 1, class 2 and class 3. MHC proteins have immunoglobulin-like structure. First, we will see class 1 molecules. MHC1 occurs as alpha chain composed of three domains: alpha 1, alpha 2, alpha 3. The alpha 1 rests upon a unit of non-MHC molecule beta 2 microglobulin, which is encoded on human chromosome 15. The alpha 3 subunit is transmembrane, anchoring the MHC class 1 molecule to the cell membrane. The peptide being presented is held by the floor of the peptide binding groove in the central region of alpha 1 alpha 2 heterodimer, a molecule composed of two non-identical subunits. The genetically encoded and expressed sequence of amino acids, the sequence of residues of the peptide binding groove's floor determines which particular peptide residues it binds. Next is class 2 molecule. MHC class 2 is formed of two chains alpha and beta, each having two domains alpha 1 and alpha 2 and beta 1 and beta 2, each chain having a transmembrane domain alpha 1 and beta 2 respectively, anchoring the MHC class 2 molecule to the cell membrane. The peptide binding groove is formed of the heterodimer of alpha 1 and beta 1. MHC class 2 molecules in humans have 5 to 6 isotypes. Classic molecules present peptides to CD4 plus lymphocytes. Non-classic molecules, accessories with intracellular functions are not exposed on cell membrane. But in internal membranes in lysosomes, normally loading the antigenic peptide onto classical MHC class 2 molecules. Next is class 3 molecule. Class 3 molecules have physiological roles unlike class 1 and class 2, but are encoded between them in the short arm of human chromosome 6. Class 3 molecules include several secreted proteins with immune functions like components of the complement system, cytokines and heat shock proteins. The complement system consists of a number of small proteins found in the blood, generally synthesized by the liver and normally circulating as inactive precursors which are called proproteins. When stimulated by one of several triggers, proteases in the system cleaves specific proteins to release cytokines and initiate an amplifying cascade of further cleavages. The end result of this activation cascade is massive amplification of the response and activation of the cell killing membrane attack complex. Over 25 proteins and protein fragments make up the complement system, including serum proteins, serocell proteins and cell membrane receptors. They count for about 5% of the globulin fraction of blood serum. Thus, the immune system is a network of cells, tissues and organs that work together to defend the body against attacks by foreign invaders. The human body provides an ideal environment for many microbes. It is the immune system's job to keep them out or failing that to seek out and destroy them.